Digestive glands. The digestive glands associated with the alimentary canal include the salivary glands, the liver, and the pancreas. Saliva is mainly produced by three pairs of salivary glands: the parotid cheek, the submaxillary oblique, submandibular lower jaw, and the sublinguals below the tongue. These glands, situated just outside the buccal cavity, secrete salivary juice into the buccal cavity. Liver is the largest gland of the body, weighing about 1.2 to 1.5 kg in an adult human. It is situated in the abdominal cavity, just below the diaphragm, and has two lobes. The hepatic lobules are the structural and functional unit of liver, containing hepatic cells arranged in the form of cords. Each lobule is covered by a thin connective tissue sheath called the Gleason's capsule. The bile secreted by a hepatic cells passes through the hepatic ducts, and it is stored and concentrated in a thin muscular sac called the gallbladder. The duct of gallbladder cystic duct along with the hepatic duct from the liver forms the common bile duct, figure 16.6. The bile duct and the pancreatic duct open together into the duodenum as the common hepatopancreatic duct, which is guarded by a sphincter called the sphincter of Oddi. The pancreas is a compound, both exocrine and endocrine, elongated organ situated between the limbs of the C-shaped duodenum. The exocrine portion secrete an alkaline pancreatic juice containing enzymes, and the endocrine portion secrete hormones, insulin, and glucagon.
Digestion of food. The process of digestion is accomplished by mechanical and chemical processes. The buccal cavity performs two major functions: mastication of food and facilitation of swallowing. The teeth and the tongue, with the help of saliva, masticate and mix up the food thoroughly. Mucus in saliva helps in lubricating and adhering the masticated food particles into a bolus. The bolus is then conveyed into the pharynx and then into the esophagus by swallowing or deglutition. The bolus further passes down through the esophagus by successive waves of muscular contraction called peristalsis. The gastroesophageal sphincter controls the passage of food into the stomach. The saliva secreted into the oral cavity contains electrolytes Na+, K+, Cl-, HCO3-, and enzymes salivary amylase and lysozyme. The chemical process of digestion is initiated into the oral cavity by the hydrolytic action of the carbohydrate splitting enzyme, the salivary amylase. About 30% of starch is hydrolyzed here by this enzyme. Optimum pH 6.8 into a disaccharide maltose. Lysozyme present in saliva act as an antibacterial agent that prevent infections. Starch, in the presence of salivary amylase under pH of 6.8, gives out maltose. The mucosa of stomach has gastric glands. Gastric glands have three major types of cells, namely: first is mucous neck cells, which secrete mucus; second, peptic or cheap cells, which secrete the proenzyme pepsinogen; and third, parietal or oxyntic cells, which secrete HCl and intrinsic factor essential for absorption of vitamin B12. The stomach stores the food for 4 to 5 hours. The food mixes thoroughly with the acidic gastric juice of the stomach by the churning movements of its muscular wall and is called the chyme. The proenzyme pepsinogen, on exposure of hydrochloric acid, get converted into the active enzyme pepsin, the proteolytic enzyme of the stomach. Pepsin converts proteins into proteoses and peptide. The mucus and bicarbonates present in the gastric juice play an important role in lubrication and protection of the mucosal epithelium from excoriation by the highly concentrated hydrochloric acid. HCl provides the acidic pH 1.8 optimal for pepsins. Rennins is a proteolytic enzyme found in gastric juice of infants which help in the digestion of milk proteins. Small amount of lipases are also secreted by gastric glands. Various types of movements are generated by the muscularis layer of the small intestine. These movements help in through mixing up of the food with various secretion in the intestine and thereby facilitate digestion. The bile, pancreatic juice, and the intestinal juice are the secretion released into the small intestine. Pancreatic juice and bile are released through the hepatopancreatic duct. The pancreatic juice contains inactive enzymes trypsinogen, chymotrypsinogen, procarboxypeptidases, amylases, lipases, and nucleases. Trypsinogen is activated by an enzyme introkinase secreted by the intestinal mucosa into active trypsin, which in turn activates the other enzymes in the pancreatic juice. The bile released into the duodenum contains bile pigments, bilirubin, and biliverdin, bile salts, cholesterol, and phospholipids, but no enzyme. Bile helps in emulsification of fat, breaking down of the fat into very small micelles. Bile also activates lipases. The intestinal mucosal epithelium has goblet cells, which secrete mucus. The secretion of the brush border cells of the mucosa along with the secretion of the goblet cells constitute the intestinal juice or succus intricus. The juice contains a variety of enzymes like disaccharidases, maltase, dipeptidases, lipases, nucleosidases, etc. The mucus along with the bicarbonates from the pancreas protect the intestinal mucosa from acid as well as provide an alkaline medium pH 7.8 for enzymatic activities. Some mucosal glands, Werner's glands also help in this.
Proteins, proteases, and peptones, partially hydrolyzed proteins, in the chyme reaching the intestine are acted upon by the proteolytic enzymes of pancreatic juice as given below. Proteins, peptones, proteases, in the presence of trypsin, chymotrypsin, and carboxypeptidases gives out dipeptidases. Carbohydrate in the chyme are hydrolyzed by pancreatic amylase into disaccharidases. Polysaccharides starch in the presence of amylase gives out disaccharides. Fats are broken down by lipases with the help of bile into di and monoglycerides. Fats in the presence of lipases, disaccharides gives out monosaccharides. Nucleases in the pancreatic juice act on nucleic acid to form nucleotides and nucleosides. Nucleic acids, the presence of nucleases, gives out nucleotides and nucleosides. The enzyme in the succus intricus act on the end products of the above reactions to form the respective simple absorbable form. These final steps in digestion occur very close to the mucosal epithelial cells of the intestine.
Dipeptides in the presence of dipeptidases gives out amino acid. Maltose in the presence of maltase gives out glucose plus glucose. Lactose in the presence of lactase gives out glucose plus lactose. Sucrose in the presence of sucrase gives out glucose plus fructose. Nucleotides in the presence of nucleotidases gives out nucleosides. In the presence of nucleosidases gives out sugar plus base. Di and monoglycerides in the presence of lipases gives out fatty acid plus glycerol. The breakdown of biomacromolecules mentioned above occur in the duodenum region of the small intestine. The simple substances thus formed are absorbed in the jejunum and ileum region of the small intestine. The undigested and unabsorbed substances are passed on to the large intestine. No significant digestive activity occurs in the large intestine. The functions of large intestine are 1. Absorption of some water, minerals, and certain drugs. Second, segregation of mucus which help in adhering the waste undigested particles together and lubricating it for an easy passage. The undigested, unabsorbed substances called feces enter into the cecum of the large intestine through ileocecal valve, which prevents the backflow of the fecal matter. It is temporarily stored in the rectum till defecation. The activity of the gastrointestinal tract are under neural and hormonal control for proper coordination of different parts. The sight, smell, and oblique or the presence of food in the oral cavity can stimulate the secretion of saliva. Gastric and intestinal secretion are also similarly stimulated by neural signals. The muscular activities of different parts of the alimentary canal can also be moderated by neural mechanisms, both local and through CNS. Hormonal control of the secretion of digestive juices is carried out by local hormones produced by the gastric and intestinal mucosa.